we realized that the best way to do a better AI was to use living neurons. Biological processors are made of living cells. This has never been done in the history. This is basically what is running ChatGPT, for instance. It was taking several kilowatts of power. Your brain is 20 watts for 100 billion of neurons. We used more sophisticated versions of these artificial neurons.
What's about the ethical issue?
The picturesque town of Vevey, Switzerland, blends natural landscapes with urban fabric. It's home to the headquarters of one of the most visionary biotech companies working to create a so-called living computer. Is it possible to combine the computational power of computers with the efficiency of the human brain? FinalSpark is a Swiss biocomputing startup that is the first in the world to build a living processor, creating a bioprocessor capable of learning while consuming over 1 million times less energy than conventional silicon systems.
What is a human brain organoid?
It's basically a small part of nervous tissue made of neurons coming from human beings.
What is it, your human brain project?
So, first, when you talk about human brain project, this is first a European project, okay, that somehow inspired us, okay, but it's not really a project related to human brain actually. There are many people doing human brain organoids in the world and in Europe also. This is not, as such, this is not something even I would say very advanced, okay. What is a bit special in our company is that what we are trained to do with these brain organoids is to use them for computation. So it's the goal that we have and the work that we do which is original.
You see what's behind the corner?
Well, I don't know if I see what's behind the corner, but that's right, that it's a very original thinking to think that human neurons can be used for computation.
How do you collect human cells?
Okay, we buy human cells from companies that are specialized to produce them, and they are pluripotent stem cells. So they come from fibroblasts, so skin cells that are reprogrammed to become pluripotent. So this means that they can become whatever type of cells.
How is it possible to create the neuron cells from using, skin cells?
Yeah, exactly. It's because there are methodologies that we can use to de-differentiate the skin cells, so bring them back to the pluripotent state, and then from this point we can differentiate them in neurons, so give them specific molecules in the culture medium from pluripotent stem cells, and then we make sure that they are pluripotent, so we check some markers to make sure that they will be able to differentiate in neurons and astrocytes and other type of cells in the brain. And then we culture them with special molecules that will induce the cells, will tell the cells to become a neuron. So we are in the cell culture room, and it's the place where we grow the brain organoids, and they are in the incubator where we found precise temperature and CO2 concentration. Inside of these plates we have different type of organoids that we are growing. First of all, actually we grow them in a special plate that allow them to create a 3D structure, and then step by step we give different type of reagents or molecules, as we can say, that will tell them to become specific type of cells, and also they will reorganize in structures that are similar to part of the brains. the function will be similar to fetal brain. Some are in the orbital shaker because the shaking helps the nutrients and the oxygens to enter inside the organoid.
How do you keep your neurons cells alive?
So we give them a special solution. So it's called cell culture medium that contains nutrients and that help them to live.
How do you keep this organoid alive in that system?
Alors, as you can see, there is maybe not really in the picture, but we have a fluidic system below. So we have pipes and bring the medium actually. So we have pump, and so the medium is really flowing below the organoid. So the organoid exactly will be on this on the top of.
So all the time they are feeding.
Yeah, all the time, yes. So with this system we can keep them alive. Usually 1, 2, 3. It can be months sometimes. But mostly it's like 2 weeks. You can see the pump speed actually of all the MEA. So for example, you can see that we are running at around 30 or 50 microliter per minute.
How long time you can keep these cells alive? 1 year, 2 years, 3 years?
We managed to keep them almost 3 years alive.
How we can connect that organoid with your neuron platform?
We put the organoid on top of the electrodes and we record their activities.
What we can see?
So this actually it's true. When you go to the website, you can see the live actually. So these pictures. So we have 4 different MEA. So actually it's a close-up on this part.
Okay, MEA is?
Multi-electrode array.
Okay.
So because there is a lot of different electrodes that you can see maybe better on this picture, you see this little. What you can see, it's we have 5 MEA. So the black one is a new one that you are trying now. But what you can see on the website is that one, the clear one. You see this one on the bottom. The 2 on the bottom. So different. So that one, it means you put 1 or 2 organoid here. So you can record from 8 electrodes. They will be independent. We call this, it's a membrane actually with a hole in the middle. We call it confetti, but well, it's just a membrane. Actually, it helps for me to put in the middle the organoid, and also it helps to reduce the noise.
Why did you start this project? Why biocomputing? Why so futuristic vision of the reality?
The first thing, I'm an engineer, so I always try to solve problems, okay, and take the best solution for a given problem, okay. And initially, when we started to work on this, I have a PhD in signal processing, basically mathematics, nothing to do with neurobiology. Okay, so I can tell you, if I could have avoided to learn an entire new field, I would have preferred. But when we started the company, we worked a lot with artificial neural networks. Artificial neural networks are simulations of neurons. This is basically what is running ChatGPT, for instance, okay, and all generative AI. And we used more sophisticated versions of these artificial neurons. And they were consuming so much energy that we realized it would be impossible to scale this. Like with 100 simulated neurons, it was taking several kilowatts of power. Imagine, your brain is 20 watts for 100 billion of neurons.
86 billion neurons we have in a human brain.
Yeah.
Using 20 watts for calculation, that's the reason that you decided to open that company?
No, the main reason of opening this company was first to do fundamental research in AI. And in the course of this fundamental research, we realized that the best way to do a better AI was to use living neurons.
What's the difference between biological and classical processor?
Between biological and classical processor, well, biological processors are made of living cells, which is totally disruptive indeed. This has never been done in the history.
I know that I saw your butterfly experiment. What is it?
Yeah, the butterfly experiment is a way to show in the browser, internet browser of a user, how he can interact directly from his internet browser in 3D with a brain organoid sitting in Switzerland in our lab. This is a visual illustration of this, and you see this butterfly flying in 3 dimensions. You can actually control its direction.
The idea is when the butterfly see the light, so it's these big blue dots, you send stimulation, and if the butterfly reply to the stimulation, you go straight, so it means you go to the light, and if not, so you are randomly going everywhere. And when he replies, so you can see actually here that after each stimulation, this big line, you can see that there is some reply. Probably a lot of neurons are discussing when you saw this big wave, actually we call burst, because there are probably more than one neuron that are sending information. So it's essentially here we see a lot of communication.
You have a communication with organoids, right?
Yeah, yes, exactly. We have now, but not between each of them. So, like, for example, here you see again a burst of a lot of neurons.
They are processing the information.
Yes.
So how do you enforce such neurons to process such information?
Yes,
Such
yes.
Dopamine?
So in the way you speak, the words that you use here makes a lot of sense to me. Okay, if you talk about processing information and enforcing a behavior, okay, so this is called learning, okay, enforcing a behavior, okay, and this is the main challenge that every researcher in the world of biocomputing has to challenge today. Okay, so your question. So one way that we are indeed pursuing here is to use, for instance, dopamine to reward the brain organoid if it does what we expect. But there are other strategies which are used by others.
Do you have any progress?
Very slow progress, okay, because we need to confirm experiments. Even if something works once, it doesn't mean it works at all, okay? We are engineers more than scientists in the sense that we want something that works.
What's about the ethical issue?
So, first we are scientists, okay? Yeah. Any questions about science, I may be able to answer. Probably, actually not. Most of the important questions in science we are not able to answer, but some of the technical questions I may be answer. Now, if you think about ethical questions, this is really out of the field of expertise of FinalSpark. And for this, we recognize this is a concern. And what we did is that we made a number of contacts with universities and ethicians, because they are experts in this field of ethics. This is their job actually to study. And what we do is that we went also to conference in November last year, for instance, to present in an ethics conference to ethicians what we were doing, so that those people who are experts in this can start to think what would be the implication of using living neurons for computation.
Why human cells?
Yeah, that's a very good question actually. If you think about computation, probably rat cells could also work very well, okay? The thing is, when you create a startup, you'll never know what are going to be the outcome. Sometimes you may discover something along the way that is very useful. And one of the things that we could discover along the way could have therapeutic applications. And I'd better have therapeutic applications for human beings than for rats.
Moment that shocked you.
that you that surprised you positively here in FinalSpark? Well, when we were able to record some activity from neurons, it was already a fantastic thing. When we were able to tow cells, from liquid nitrogen and multiply them ourselves, we were able to see some reaction of the nervous tissue to dopamine and
The waves.
Yeah. We saw some activity which is typical induced by the release of dopamine. This was one I would say major achievement for us, okay, but just always put in perspective that we were starting from ground zero.
Do you really believe that biocomputing is the future of data processing, is the future of computing?
Yes, absolutely. Specifically for one type of computing, okay? Computing of AI. Okay, like quantum computing may be restricted to some types of computing. Biocomputing will be restricted also to some tasks of computing. But AI is absolutely perfect. Then for an obvious reason, AI is based on simulations of neurons.